Assalamualaikum and a very good morning, IB, to Dr. Ibrahim and all my fellow friends. So today, Group 2 will be presenting about ultraviolet visible spectroscopy. So before we go in depth about this topic, allow me to introduce my group members. So firstly, we have Mr. Muhammad Guran. Next, we have me, Miss Iza Farzana. Then we also have Miss Nur Fatin Inani, and we also have Miss Nurul Natasha. In this presentation, we choose to use the 5W concept, which is what, how, and when. So for the first part in this presentation, I will be presenting about the introduction of UV visible spectroscopy. Next, Ms. Fatin Inani will explain about the technique on the instrumentation and how UV visible spectroscopy works. And Ms. Nurul Natasha will be continue to explain more about the formula of absorbance, the type samples that can be used, and explain more about the Beer-Lambert law. Last but not least, Mr. Guran Hashim will be explaining about the application of UV visible spectroscopy. This part, I will explain about the introduction of UV visible spectroscopy. So what actually is an UV visible spectroscopy? So UV visible spectroscopy is an analytical technique that I use to measure the absorbance of light by a chemical substance across the ultraviolet and visible range in the electromagnetic spectrum. As we know, in the electromagnetic spectrum consists of a wide range of spectrum from X-rays to radio wave. But however, in the UV visible spectroscopy, it only focused on the ultraviolet and visible range of spectrum. Next, by using UV visible spectroscopy, we can measure the light intensity that passes through a sample. And this quantitative measurement of light absorption is called spectrophotometry, where we use wavelength as a function. In UV visible spectroscopy, the wavelength range that are typically used are around ultraviolet region and visible region, where ultraviolet region covers around 160 nanometers to 375 nanometers, while in the visible region, it covers around 350 nanometers to 2500 nanometers. Ultraviolet visible spectroscopy, also known as electronic spectroscopy. This is because the principle of this kind of spectroscopy involve the transition of electrons, where the excitation of electrons occurs from lower energy levels to higher energy levels. So the picture on the left side shows the example 2 of UV visible spectroscopy, which we call the instrument as spectrophotometer. So in the right side of the picture, it shows that in the electromagnetic radiation, UV visible spectroscopy usually covers around ultraviolet radiation and also visible radiation. In the visible region, it consists of 7 colors. So now I want to share with all of you what I've learned back in my high school on how I can remember easily the 7 colors consist in the visible spectrum. I use the acronyms called R, O, Y, G, B, I, and V, Roy G, B. But before that, excuse me, I am sorry to use Malay. My acronym sounds like this. R stands for Raju, O stands for Orang, Y stands for Yang, G stands for Garang, BI stands for Bagai, and lastly V stands for Vampire. So Raju, Orang, Yang, Garang, Bagai, Vampire. So of course, R stands for red, O stands for orange, Y is yellow, G is green, B is blue, I is indigo, and lastly V is violet. And you can see in this electromagnetic spectrum, the violet color has the shortest wavelength compared to red, which have the longest wavelength in the visible spectrum. For your information, the colors consist in the visible spectrum is the colors which can be seen by human naked eye. For example, we can see the blue color of iron copper solution. Next, I will pass the presentation to Ms. Fatin Inani.
Hi, I am Nani. Next, I will continue the second part of this presentation, which is regarding the instruments and how it is works. So in this part, I will be explain about the instruments and how all the instruments working, which is the technique. And after that, Tasha will explain about the absorbance and Beer-Lambert law. And the last one is how to interpret the graph. So before I introducing to all of you the instrument that have in the spectroscopy, let us rewind back the basic principle in the UV visible spectroscopy. So UV visible spectroscopy is based on phenomenon of electronic excitation, which is when a UV light radiation is passing through a sample, yeah, the atoms or molecule in the sample will absorb the energy and move from the ground state to the excited state. So, and if it occurs, the residual radiation, when it is passed through a prism, which is one of the types of monochromator, is a spectrum with gap in it, and it is called an absorption spectrum. Thus, the excitation is detected by the detector. Here, there will be a detector, and being recorded. Let's move on to the instruments of the spectrometer. They will have 7 types of basic instrument that you have to be remember, which is the light source, monochrometer, beam splitter, sample, reference, detectors, ratios, and also the output spectrum. So what is the function each of them? The deuterium lamp is used to detect the UV region, which is when the sample that have the wavelength around the range 160 until 375 nanometer. And for the tungsten lamp, is being used to detect the visible region when the sample that have the wavelength around 350 to 2500 2500 nanometer. Now, let's move to the second types of instrument, which is the monochrometer. So monochrometer is a device used to resolve wide band of the polychromatic light radiation into a narrow band of monochromatic radiation. Here we'll have 3 types of monochrometer, which is the prism, filters, and the gratings. As you can see from the figure here, this is one of the type of monochrometer, which is the prism. As the white light passing through the prism, they will strike into 7 types of colors, which is the rainbow's color in the white band into a narrow band radiation. So next is the beam splitter. Beam splitter is used to split the incident light at a designated ratio in 2 separate beams, which is the reference beam and also the sample beams. Number 4 is the sample. Sample is a small transparent container cuvette containing a solution of the compound being studied in a transparent solvent. And number 5 is the reference. Reference is an identical cuvette containing only the solvent. Here, you need to understand that for the sample, we only use the colorless or transparent solvent in the cuvette. But for the reference, we will use the color solvent, which commonly we will use the compound that have from the transition metal group. Okay. Now, number 6 is detector. Detector is used to convert light energy into electrical signal that are displayed on a read of devices. And number 7 is output spectrum, where the ratios of the intensity from the beam and from the reference beam and the sample beam will be sent to the computer, and the wavelength can be identified, and from here we can know what is the complexometric color that can be obtained. So for now, I've been explaining to all of you the basic instrument and the function all of them. So now let's move on to the how does it working. First is the beam of light. So when the beam of light is being turned on either from the light sources UV or from the visible, it will be reflected at the mirror one, and the UV radiation light will passing through the slit one. The slit one here is used to ensure all of these light photons are traveling along parallel pathways so that when they strike at the diffraction grating, which is one of the type of monochrometer, they will be refracted into a rainbow color, 7 rainbow colors. So each of wavelength of light is moving through a different place in space. So only one type of wavelength of light in this situation is going to make it through the second slit and into the filter. One single wavelength will passing through until it reflects at the second mirror at the mirror 2, and it will be separated at the half mirror, or can be called or can be said it will be split at the half mirror. Half mirror here also be known as the beam splitter. So two types of beam have been splitted into the sample beam and also the reference beam. So the first beam is the sample beam. The sample beam passed through a sample transparent container, which is a cuvette, containing a solution of the compounds being studied in a transparent solvent, while the other beam, which is the reference beam, passing through an identical cuvette containing only the solvent. So the intensity of these light beams are then measured by the electronic detectors and compared. The intensity of the reference beam, which should have suffered little or no light absorption, is defined as I naught, while the beam or the intensity of the sample beam is defined as I. So we will obtain the ratio from the sample beam and the reference beam, And from here, the spectrometer automatically will scan all the component wavelength in the manner described. So the ultraviolet region scan is normally from 160 until 275 nanometer, and the visible portion is from 350 until 2500 nanometer. So this is the sample of the wavelength that can be obtained. Um, the absorbance will give the value of the wavelength, and with the value of a wavelength we can determine what the complete symmetric color that will visible to our eyes. We can use the artist color wheel. This is the artist color wheel. So the color that have been absorbed is the color, the opposite color is the color that have been visible to our eyes. So for example, if the range of our wavelength is around 400 until 430, so the color that have been absorbed is violet, but the color that have been scattered or been visible to our eyes is the yellow color, which is the opposite one.
So now let's move on to the calculation part. This is the formula of intensity of absorption, which is A equal to log capital I null over I. So A here stands for absorbance, while I here is stand for intensity. For I null is incident intensity, which is intensity before light pass through the sample in the cuvette. So I here is intensity after the light is passing through the sample in the cuvette. So the sample does not absorb light of given wavelength if intensity here is equal with incident intensity. So we can conclude that there is no absorption occur because log 1 is equal to 0. So A here will be 0. So we can get the value of A if incident intensity, the value of incident intensity here is higher than intensity. So if you just insert any value inside this calculation, then we can get a positive value. Therefore, we can get a graph of absorbance versus wavelength. What will happen if intensity, the value of intensity here is higher than incident intensity? Then if we just put any value inside this equation, which intensity is higher than incident intensity, therefore we will get a negative value. It means that there is no sample present inside the spectrophotometer. So if there is no absorption here, it means that the transmittance will be 1. So this is the formula of Beer-Lambert law. Beer-Lambert law is a way to measure the concentration of solution based on how the sample or analyte absorb the light. So it is used to relate the absorbent to the molar concentration J of the absorbing species J and the optical path length. So this is the formula of Beer-Lambert law, which is equal to epsilon multiply with concentration and also optical path length. So A here, which is absorbent, where we can get the certain amount of wavelength that been absorbed by the sample. For epsilon, which is molar absorption coefficient, it will tell us how well the molecule absorb the wavelength. So if the value of epsilon here is high, it means that the molecule will absorb more light. So for molar concentration of the absorbing species here, it will tell us how many molecules are in a given volume. So when the value of when molar concentration is high, it means that there are more molecules in the sample will absorb the light. So for optical path length here, it will affect how many molecules light encounter on its journey. So if the higher optical path length, the tendency of light to meet with the molecule is also high. Okay, so this is the formula of intensity of absorption, and this is formula of Beer-Lambert law. So both of this equation is equal to A. So if you can see from this diagram, this is incident intensity, which is intensity before the light is passing through the sample, and this is intensity after the light is passing through the sample, and this is optical path length. And inside this cuvette is our sample that contain our analyte. So next, okay, this is the graph. The first graph here is the rough data that we got from UV visible spectrophotometer, which is graph of absorbance versus wavelength in nanometer. So this is only an example where I will show you how to read the graph here. So if you can see, the peak here is at 520 nanometer. So around plus minus 520, which is between 490 nanometer and 560 nanometer here. So it means that the analyte will absorb green color, emit red color. It means that the red color will appear. So the absorbance here is 1.2. So for the second graph here, it is a graph of absorbance against concentration, which is Beer-Lambert law graph. So if you can see the pattern of this graph, it is a linear graph. So from this graph, actually you can find the unknown concentration. Whereas if you guys know that for linear graph, it is equal to mx. C. So how to get the value of y, m, and x? So refer to the previous slide. Log of the formula of intensity of absorption here will be our y, and epicenter here will be our m, which is the slope, and concentration here will be our x. Okay, so the value of y can be obtained from this equation, and then we have to find the unknown concentration, which is x, from this graph. So maybe you can get the value of y, any value here, and then you just have to find the concentration, the unknown concentration from this graph. So this is where the unknown concentration is. Okay, so next is the sample, where there is suitable sample that can be used, and there's also non-suitable sample that could not be used in UV visible spectrophotometer. So first, the suitable sample here, it need to dissolve in solvent like water or any organic solvent like ethanol and hexane. Second, it need to absorb UV visible light. So if the sample does not absorb UV or visible light, therefore there is no absorption occur, and it will be 0. So there will be no data. So lastly, it has different molar absorptivity constant. So for this statement, this apply when there is more than one analyte in a solution. In other words, from this, due to different molar absorptivity constant, we can do quantitative measurement on the analyte. So for non-suitable sample, the analyte that have a photochemical reaction at or above the wavelength range of interference. So like what Iza and Nani already say, the wavelength range is around 160 until 2500 nanometers. So if wavelength is at 2500 or above than that, therefore it is unsuitable sample. Next, the sample that unclear or colloidal sample. So this is one of the example of inorganic compound that we can use to analyze with the UV visible spectrophotometer, which is tetraamine copper 2 sulfate pentrahydride.
Hello, in this part I will talk about application of UV visible spectroscopy. There are 4 applications of UV visible spectroscopy. First, detection of impurities. Second, spectrophotometric detection. Third, chemical kinetics. And lastly, determination of configuration of geometrical isomers. First, detection of impurities. UV visible spectroscopy is the best method for determining impurity organic compound. The presence of impurity in the compound will give additional peak. As you can see the right side right now, UV spectra of paracetamol. There are 2 types of spectra. At the top, standard PCM, poor PCM, and at the below, PCM with impurity. Compared to types of spectra, you can see the difference between these. As you can see right now, black circle have additional peaks. So we can determine that at the below spectra have impurities in the compound. Other than that, we can determine impurities by measure the absorbance at specific wavelength. For example, benzene always appears as common impurity in cyclohexene. We can check by detect by absorption at 25.5 nanometer. Second application, spectrophotometric detection. To undergo spectrophotometric detection, either titrant or reaction product must absorb the radiation. If not, cannot undergo the spectrophotometric detection. The chance on absorption can be used to determine the endpoint. These titration are based on First law. As we understand, First law explains that the relation between absorbance and concentration is directly proportional. As you can see right now, there are 6 types of draft titration. Why? Because the draft can be determined by depends what the substance absorb the radiation. For example, graph A, only the titles absorb. In this graph, you can see the red arrow. The red arrow is determining the endpoint. It shows where the graph is discontinuous. Okay. Third application, chemical kinetics. We can use UV visible spectra monitoring. By use this, we can monitor slow reaction with half-life of hours or even days. Other than that, we can use stop flow techniques when we can study fast reaction where half-life of between 5 milliseconds and 10 seconds. As you can see at the right side now, this is the type of structure of stopper flow distortion. When the reaction occurs, can be monitored at single wavelength, or successive spectra can be measured very rapidly by using a diode array detector. And lastly, slide, we talk about determination of configuration of geometrical isomer. There are 2 types isomer at the right side, isomer A, isomer B. Isomer A show the cheese formation, cheese form, while B transform. As you can see, isomer A have the lower wavelength compared to isomer B, which are 283 nanometer and 296 nanometer. Why isomer A have lower wavelength is due to steric hindrance. As we know, steric hindrance destabilize the compound. That means energy of the molecule will increase while the wavelength will be decreased. That's all for me.
So now, let's watch a video on the summary of what had been presented by Ms. Fatin Inani and also Ms. Nurul Natasha about the technique of UV visible spectroscopy.
Ultraviolet visible spectrometry tells us about electronic transitions in atoms and molecules. Spectra are produced when electrons in molecules or atoms move from one electronic energy level to another of higher energy. In doing so, they absorb energy equal to the gap between the 2 levels. Compounds that absorb in the visible region, such as some transition metal compounds and organic dyes, are coloured. Those that absorb only in the ultraviolet region are colourless. Inside a UV visible spectrometer, there are usually 2 light sources, one giving out visible light and one ultraviolet. This one is a tungsten lamp, like a car headlamp bulb, for the visible region, and this deuterium lamp gives out ultraviolet. This mirror directs light from the appropriate source into the monochromator. This contains a diffraction grating that acts rather like the plane surface of a CD to split the light into its constituent wavelengths. Different wavelengths correspond to different colours. Red is about 700 nanometers and blue around 400 nanometers. Wavelengths shorter than about 350 nanometers are called ultraviolet. Shorter wavelength light has higher energy. The source produces white light that includes all wavelengths, all colours. The instrument scans through the spectrum, sending different wavelengths of light through the sample in sequence. This is done by the grating, which rotates. A single wavelength passes into the modulator, which consists of a rotor with mirrors on it. This chops the light into 2 beams. One beam passes through the sample cell, while the other passes through the reference cell. So the instrument is referred to as a double beam instrument. Both sample and reference beams are directed by mirrors onto a detector. This compares their intensities and sends a signal proportional to the ratio of their intensities to the computer that controls the instrument. The logarithm of this ratio gives a quantity called absorbance, which is a measure of how much light has been absorbed by the sample at that particular wavelength. Ultraviolet visible spectra are usually run on solutions. Light does not normally pass through solid samples. Here, we will run the spectrum of a green food dye. To run the spectrum, we place some of the solvent in a sample cuvette to act as a blank, a reference. The cuvettes may be made of glass or plastic if only the visible region of the spectrum is required. Quartz cuvettes are needed for work in the ultraviolet range because glass and plastics absorb UV light. We then place a solution of the sample in a second cuvette. The blank and the sample are placed in the sample holders. The lid is closed to prevent light from the laboratory interfering with the spectrum. The operator types details, such as the wavelength range required and scanning speed, into the computer that controls the instrument. He also zeroes the instrument at a point where the sample does not absorb, and then starts the scan. The spectrum appears on screen. The data is saved on the computer and a hard copy can be printed. The horizontal axis is normally wavelength, and the vertical one absorbance, which is a measure of the amount of light absorbed by the sample. The peaks on the spectrum are the wavelengths of light that are absorbed by the sample, and the troughs are where light passes through. So the dye absorbs orange and blue light and lets through green, so it appears green in colour.
That's all from our group presentation. So after this, if any of you might have questions, you may ask. Thank you.
Thank you.
Thank you.